Thank you very much. I would like to present our work on Apobec family of enzyme, and we studied 3D localization of Apobec Induced Mutation using high-C data. So already several years we are studying Apobec family enzyme because it was found earlier that this family of enzyme is implicated in cancer mutagenesis. So it was obvious in the beginning of the era of sequencing of cancer genomes that the researchers found some cluster of mutation with the so-called Apobec signature in DNA. And normally, Apobec family enzyme is a part of the immune system, where active against viruses and retrotransposons, and they can Apobec enzymes can bind to the single-stranded DNA. But as researchers found these mutations in human double-stranded DNA, there is an opinion that in some processes, human double-stranded DNA can unwind and or occur double-stranded damage breaks, and in that moment, Apobec can bind to the single-stranded DNA and mutate it. So our previous studies was related to the find in which particular processes Apobec can mutate single-stranded DNA. So we studied correlation of Apobec mutagenesis with replication and found big correlations. We also studied connection of transcription and Apobec mutagenesis and found a little bit less, but also correlations. And we think it is connected with the formation of the R loops. And surprisingly, we did not found any increase of the Apobec mutagenesis in non-canonical DNA structures, although these structures contain big stretches of single-stranded DNA. So the Apobec mutate cytosine into thymine or guanine, and its specificity shown on this sequence logo. So upstream of cytosines, usually we can find thymine, and downstream position from the cytosine is usually adenine or thymine. Here you can see the so-called Apobec mutational signatures. When the researchers studied mutations in cancer genomes, they can found the different so-called mutational signatures, and this is two signatures from so-called cosmic database, and signature 2 and signature 13 is associated with the Apobec activity. Here you can see that there are high probability of mutation of cytosine into guanine and thymine. So what is interesting about Apobec mutagenesis, that researchers found that this has a episodic activity. So when the some cell lines was sequenced, the researchers found only episodic activity of Apobec mutagenesis, and it doesn't clear what is associated with this burst of Apobec activity. And recently, Apobec was associated with the drug resistance to anti-cancer therapy. So in this paper, the researchers found that therapy with the tyrosine kinase inhibitors associated with the activity of Apobec enzymes, and this Apobec activity allows cancer cells acquire drug resistance. Here I will show some slides where you can see that Apobec induced mutations frequently located in the DNA as clusters. So in this so-called rainfall plot, you can see some clusters on different chromosomes, and it's usually associated that Apobec can bind and slide along the DNA to form these clusters of mutations. And to start these projects, we were interested in what about the other scattered mutations about which are scattered along the genome. We had a hypothesis that these mutations may be also clustered, but in 3D. So the goal of our project was to analyze the 3D distribution of Apobec induced mutations in using high-C data in cancer samples. So what is high-C data? High-C data is the method which allows to find 3D association of different DNA loci. So it can allow cross-linking of DNA closely located in 3D space, and then sequence it and found the association between the different regions of DNA in 3D space. So this is a slide about methods that we applied. So we took the somatic mutations from famous Peacock projects, and we took not directly the high-C data, but we took already constructed three-dimensional chromatin models from GCDB database. And to select the cancer samples with the Apobec activity, we applied a metric introduced by Dmitry Gordenin group, so-called Apobec enrichment, that calculate the fraction of mutation with the Apobec signature relative to the any mutations in cytosines. So we selected samples with the Apobec enrichment higher than the 2.0 value, and we applied only for the cancer types which is known for Apobec activity. This is the breast cancer, lung cancer, head and neck cancer, cervical cancer, and esophageal carcinoma. So to analyze scattered mutations, we first excluded one-dimensional mutation clusters. So we analyzed the distance between mutations along the DNA. We defined the clusters of mutations and excluded from the analysis. Then they used model from GCDB database, and this model allowed to find the 3D coordinates for each genome regions. So using these three-dimensional models, we can calculate a distance between the pairs of mutations, for example, and we estimated a tendency of mutations to form clusters in 3D. We applied for this so-called Hopkins statistic. This is metric that compare the distances between actual mutations and randomly generated data points in 3D space, and allows to estimate whether the mutations form clusters in 3D space. So here our results on the x-axis. You see the this metric that I called about. It's called Apobec enrichment, and on the y-axis you can see values of Hopkins statistics that describe the clusterization of mutations in 3D space. So the Apobec active samples actually located on the right part of this plot, and samples with a little number of Apobec induced mutations here on the left slide. So you can see the samples with a low Apobec activity have a very clustered mutations. This can explain it because it is known that usually in cancer mutagenesis, mutations located mostly in the exterior of the nucleus, in so-called lamina-associated regions. So they form clusters there. But for Apobec active samples, we can see a little different picture. Some samples has a very low value of clusterizations, and some of them have some clusterization, but anyway, this value of clusterizations is much less than for samples with low Apobec activity. And on the next slide, I'm sorry, on the next slide, on this slide, you also where the size of the points associated with the number of mutations, you can see that these large bubbles of associated with the chromosome with big number of Apobec inducing mutations, and they all have usually the smaller value of the Hopkins statistic in comparison with the Apobec less active samples. So our conclusion is that we did not find any clusterization of Apobec induced mutation in 3D, but at least this is different from the background cancer mutagenesis, and it is interesting what mechanism is responsible for such a picture. So I would like to thank our team members, Gennady Ponomarev and Alexandra Denisova, who participated in this project. And thank you very much for your attention. I'm happy to answer the questions.
Thank you very much. So at the moment, I do not see any questions in the chat, but probably somebody wants to ask orally. I do not see really hands up indicators. It seems that there are no questions, but anyhow, your email address and coordinates will be available to all participants, and people can ask you directly via email. Thank you again.
And thank you very much for your attention.